Hello, my name is Raul Perez-Jimenez and I'm the group leader of the nanobiomechanics group here at CIC nanoGUNE. In our group, we study the mechanical properties of protein. Basically, what we do is apply mechanical forces to protein to study how these molecules respond to a mechanical force. We use single molecule techniques, but the first thing we need to do is basically obtain protein, and this is what we do in this laboratory. We use a standard protocol of molecular biology to prepare proteins, and the final step of the protein preparation is the site exclusion chromatographer. We are using this column, we can actually obtain the proteins. So we basically have here a number of tubes where we have been obtaining our protein. So using this protein, which are highly purified, we're going to go now and see how we perform an experiment in an actual atomic force spectrometer. Here is where we do our experiment. This is an atomic force spectrometer that basically allows us to grab literally a single protein and stretch it while we are applying a controlled mechanical force. We do that thanks to a cantilever tip that is inside of this setup, and where we actually placed our protein. So the experiment is quite simple. We grab a protein and then we stretch it by applying a force that we control. Right now we have an experiment running. This is a very simple experiment in which we apply a force of about 180 piconewtons, and we can see these three steps here, which are the unfolding of three individual proteins at that particular force. This is, as I said, a simple experiment. We can make it more complicated by quenching the force, monitoring protein folding, monitoring chemical reaction. So this machine actually allows us to study not only the physical properties of proteins, but also the chemistry of proteins under force in a very controlled way.